said here is there is a transition from stable points to oscillatory behavior, and this is one of the nicest way to do this is to pass it through a Hopf bifurcation, right. That is one of our very nice mechanisms for transitioning between these two behaviors. I am going to show you another way in which you actually get oscillations and steady states in a neuronal model. So, I am going to switch now to a Morse-Laccar of the other type. This one is going to be called the SNLC, actually I prefer SNIC, SNIC version of things. So, saddle node on a limit cycle or saddle node on an invariant circle, circle, SNIC. I like to call it a SNIC bifurcation. So, if you hear me calling it a SNIC, bear with me, it is what this is, saddle node on a limit cycle. So, here is what is going to happen. In this, do not make a difference between them. Yeah. Okay. So, let us see what is going to happen over here. We remember we made a couple of points about how you can have amplitudes that can vary in these in the previous case, right. Now, I am going to show you a case where the amplitude is going to be nearly the same. Your firing is going to be rather canonical. Your firing is going to be, you might change a little bit, but it is going to be more or less the same thing. But what can change is how long it takes to fire. You might inject a current, but you might not see anything, right. You might wait for a while, you are waiting and boom, this thing spikes, right. So, how can something like that happen? It would be very useful in a spike timing kind of code to be able to tune the spike locations, the spikes as they come, it spikes now and then it spikes then and then it spikes then. It would be very, if it is spike code, this will be very important to be able to say what is the difference between two spikes is, right, the interspike interval if you like. So, can you tune that? Can it be that your spike is not changing in height, it is not changing in amplitude, but you have control over the interspike interval, you have a control over how much it spikes. So, for that reason you would want to have neurons or behaviors in neurons where the latency to firing, as it is called, can be arbitrary. So, can we then, so we are going to now study a version of the model in which the latency is going to be the important object of our study rather than the amplitude. The amplitude is going to be nearly canonical, right. So, when a neuron is nearly canonical in terms of its shape, then you do not worry so much about that, because it is going to be just that, you worry about the timing of it, right. So, in many neuron models, for example, one actually leaves out the spike entirely. So, there are neuronal models, so instead of building the full Hodgkin-Huxley, one builds caricature models. Caricature models would be, one caricature model could be the following. I am going to write down a set of equations that says my neuron is going to grow like this, and when it reaches some threshold, this is V and this is time. If it reaches some threshold, well, reset it back to some resting potential and let it climb again, right. So, what happened to the spike? Well, you just leave it out. Okay. So, there are models, integrate and fire, quadratic integrate and fire, where you are dispensing, and the equations become very simple. You lost the whole burden of having to work with either a Hodgkin-Huxley or the Morse-Laccar or the Fitch-Nagumo. You are working with a very canonical, with a very cartoonish kind of neuron, but in the neuron does what you need it to do. It is going up, it fires, comes down, goes up, fires and comes down, goes up, fires, and you leave out all of the stuff corresponding to the spike. Okay. So, especially when you are interested in asking for the timing of codes, you do not bother about height. So, if you see neuronal models where you do not actually get a spike, now you know where it comes from. Okay. So, we will not study these, but again another project idea to study neuronal models that are of simplified types like that. There is a nice paper by Izhikevich, I hope I get his spelling right, which has a wonderful set of cartoon neurons, and perhaps that can be one of the projects you can work on to study cartoon models. By cartoon models, I don't mean to disparage them, I mean to say they are very simplified models of the neuron. Okay. They are not biophysically relevant, but they capture the essential behavior of a neuron. So, nice papers by Izhikevich on that. A lot of other people as well, but I like some of the work that comes out of Izhikevich. Eugene Izhikevich, if you like to know more, okay, and you can search up his papers. Okay, so let's talk about this. I'm going to show you a figure which I'm going to then reproduce for you, go through all of the motions to show you how this is done. Okay, so this is, I'm not deliberately not showing you the other figure because I'll show that to you in a minute. Okay, you can see spikes that are nearly canonical in terms of the shape. It's the timing that this is the variable latency. Can I use this word latency? It's the spike can emerge relatively rapidly, or after you wait a little bit, or if you wait a little longer, or if you waited a long time, the spike can emerge even though your injection of current is at the same time. Okay. So, how can this happen? So, we are going to study this from the point of view of changing initial conditions in your phase space to study trajectories that have variable latency. Okay. So, let us try and do that. Let me see if I can set it up for you. Again, we are going to okay. Let me inject a certain amount of current in it. I wonder what the current was for this. I'm trying to scroll through this to see if Bart told me what current he had used for this diagram over here. If I can't find it, I'll try and figure it myself. Okay, perhaps not. Let me start with some arbitrary current. Let me see. Minus 15. Is that, did you see that somewhere? Okay, we'll check. Okay, I'll try one of those. Thanks. So, that's my voltage graph, right. I applied 10 at the moment. I'll see where I can go with this. To see where I can go with it, I'm going to switch to my phase plane. Okay, makes it easier for me to do. I can study it through this. I can study it in the time domain. That means I can just change my current and I can see what I can do. Would you like me to do that? Should I? For those of you who are trying to do projects on this, especially, if you want me to do it, tell me quickly. I'll do it for you and show you. Okay, so I'll do this thing called initial condition and range, and I'm going to take IAP. I'm going to step it in 20 from, say, did you say minus 15 to, no, did you say 15 to 45? 15 to 40. Okay, let's go over that. 15 to 40. Let's get started. Oops, I should set up my viewport to observe all these properly. So, y min should be, say, minus 40 to good value could be 40. Oh, I should have not quite. See, this is what is involved in playing around with things. You don't quite get what you want and you change things until you get something interesting. Well, I changed. So if you there's a Moir's pattern happening over here, but there's these lines are not there. These are lines that go like that. I swept my current, just to show you they can do that also in XPP. I swept my current between 15 and 40 as was suggested in 20 steps, and I get 20 different resting potentials. Okay, so another of those things, another exercise for you to play around with this. Okay, let's go back down to a single current. I'll keep it 20 for now, or maybe 15. Okay. So I'm going to switch to a phase plane view. I'm going to take x min. Oops. Something's gone wrong. Okay, almost there. Okay, hold on, let me not do this in real time. I think I'm messing things up. Okay. I see what's going on. There it is. Okay, I managed to get it. All right, so let's see. So I've set up my viewport. As you can see, it took me a bit of effort to do it, but my current is 20, and I get 3 intersections. What was it? How many intersections did we have for the other case, for the Hopf case? You had just the one, right? Now you've got 3 intersections. Okay, so what would be our first point of analysis in trying in studying what these intersections are going to do for us? I imagine right off the bat that this guy is the one that corresponding the resting state. But what are these other ones doing? So we will have to look into that. Yes. Okay. So let's take a look into that. What should I do to study my fixed points? I should look for its stability, right? So I should probably focus on this and study the stability of that, because that's the one I think corresponds to my. Yeah, I'll do that next. First, let me check its stability, because the first thing that strikes to me is, am I really at the steady state over here, right? Yeah, because if I have a steady state over there at that value, then that can be my resting potential without any stimulation other than what I already have. Okay, so singular points, mouse, where did I already click it? Here it is. Okay, so that's my fixed point. I can see it's stable, so that's good. Okay, now let's do as was suggested. Let me plot a direction field. Oh, I did a flow, sorry. I spoiled the suspense, didn't I? Okay, there it is. Okay, so we see that clearly the field is becoming very small close to this point. There seems to be a large circular motion going on over there. So perhaps one of these will give us the trajectories that we want, right? So there's a formal way in which you can plot trajectories. You can start with a vector, move a little bit along that vector, produce the next vector at that position where you've ended up, and then plot the next one, plot the next one, plot the next one, and so on. And that corresponds to what Sitabra briefly mentioned yesterday, the Euler method of propagating. When you solve a differential equation, there is an Euler method for propagating. The phase plane version of that is to follow arrows as you move along the trajectory. Get an arrow, move a little along that arrow, get to the head of that arrow, move a little, find the next arrow at that position, find the next arrow at that position, and with this you can trace out the entire trajectory. Okay, so that's the way we think about these things. But you can sort of trace out a curve in your head. Something is happening where it's moving around and it's coming down over there. Okay, so let me show you something else now. Let me study the stability of let's study the stability of these two other points also. What could they be doing for us? Okay, so I'm going to try and do single points mouse, but I'm going to study the stability of this fellow. That actually turns out to be unstable. Okay, let's take a look at this one. Singular points, again mouse. Okay, so that also turns out to be I'll move this out of the way, turns out to be unstable. Okay, yeah, we'll get there to the flow also in a minute. Okay, so it's unstable. Okay, and it's asking me something about what these invariant sets are and so on. Okay, so let me first tell you a little bit about this the behavior over here. For those of you who know a little bit about eigenvalues, if you look at the eigenvalues corresponding to this, the positive part, there's no imaginary part, that's 0. The positive part, one of them has a positive eigenvalue, the other one has a negative eigenvalue. What this tells us is there are two directions around it, there are two vectors. If you were to be on one vector, you would move on one of those vectors, you would move in towards that fixed point, and the other vector corresponding to the positive eigenvalue, you would move out from that. So there are two directions. Along one direction you move in to the fixed point, closer to the fixed point. Along another direction you move out from the fixed point, and any other position is a combination of these. Does that make sense? Okay, that's what the invariant sets are going to show us. It will start by plotting that set of vectors, and then it will stretch those vectors out under the flow. Okay, so let's see if we can do that. That's 1, 2, 3, 4. So you'll have to look a little bit hard. There are 4 directions here, or 2 directions, sorry. This is one direction and that's another direction. Never mind this little block, okay, that's a little break over here, that's a function of the numerics. There is one direction here and one direction here. Can you guess, looking at the direction field, is this the stable eigendirection or is this the stable eigendirection? Is this the unstable eigendirection or is this the unstable eigendirection? Which one do you think? If I move along, if I stay very close to the blue, am I going to be drawn in to this point or am I going to be drawn out to this point? Yeah, you guess it from the direction flow, right? From the direction field. Yeah, that sounds right, right? So for example, this arrow over here tells me that, if I move along here, I'm actually going out. This thing, it's not very clear, but there's a little arrow here which says you're going in. Right, so you can look at the direction field and sort of guess which direction is which eigendirection is the one that's stable and which eigendirection is called the unstable one. So these are called the stable and unstable manifolds of this point. Okay, the word manifold is a misnomer over here. If anyone's interested, in knowing more details, we can discuss it offline, but these two eigendirections correspond to the stable and unstable directions, and anywhere else you will be dragged in and then dragged out under the influence of the unstable direction and the stable direction. The stable direction will grab hold of you over here for a while, then the unstable direction grabs hold of you and you move away. At the fixed point. At the fixed point there, if you are at the fixed point, you can't go anywhere, you are not going anywhere. Okay, so here is my thinking, I am trying to put it together. In the next minute I will see what the direction flow is. I am thinking that if I am somewhere here, if I was here and made a perturbation over here, then I will probably come back there. If I am perturbed to here, I will probably come back. If perturbed anywhere here, I seem to be coming back here, and that stands in well with knowing that this is a stable point, right. Okay, if I move a little bit farther away, at some point I am going to cross over onto this side, and then I am to be dragged into that direction, right. So, I have my threshold for me, there is some threshold here. Do you all see that? Okay, that is going to be my threshold of excitability, but it is a very peculiar sort of behavior, very distinct from the one that we saw previously for the following reason. Ready? If I am close to this blue line, what did you think was going to happen? We said that you will be dragged towards this fixed point, right. If you were dragged into the fixed point, what would happen? Everything would be flat, right. So, you will stay flat for a long time and then leave, and that is the origin of latency. Okay. So, you will I'm next I am going to try to do some magic with my hands, try to move my cursor close to that blue line, which I am not going to succeed at, but I want you to understand the logic of this. If I stick close to the stable manifold, I am being dragged towards the fixed point. I can't go into the fixed point because I am not on the stable manifold. So, at some point the unstable direction will take me away, right, but I will spend a lot of time hugging the blue manifold and then walk away along the yellow. If I walk away along the yellow, I have to make this huge excursion that I come back, that is my action potential, but the point time spent waiting close to the blue gives me my latency.
Is there a biological parallel to this, like in terms of what is happening in terms of ion channels?
Good question, an exercise for you to do. You can grab your trajectory, which I am going to show you how to get, and then you can work through the mechanics of it in a biophysical way by looking at the voltage and what the end gate is doing, right. Okay, so I am going to do initial conditions, mice, doesn't work. So, a trajectory like this we saw that is going to go around like that, okay, boom boom, and if I plot it over here, you will see that it is a single action potential. Maybe I should do that. So, this trajectory over here corresponds to this trajectory over here, right. Again, the latency is not terribly much. My goal is to find something which has a latency like that, which has a large enough latency that is, you wait for a couple of tens of milliseconds before you can get it. Let us see if I can do that. Okay. So, in order to do that, I am going to drag myself close to here. Okay. If I take another point close to this trajectory, I will get a trajectory that is nearly the same as this trajectory. Okay. Another exercise, plot this over and see that this it will be shifted somewhat, but it will not have a dramatically lowered amplitude, pretty much the same canonical waveform, right, because I can see that these trajectories are nearly the same, they all more or less go to the same size, right. That was the amplitude in the new case, that was the amplitude in the previous case, there is very little difference between the amplitude of these two, right. So, I have trajectories of nearly the same amplitude. Of course, the waveforms are exactly different, I mean if you compare them exactly they are different, but they are nearly the same shape, but it is the latency I am interested in. Okay, let us try another one. Okay. So, I have got a couple of trajectories I have drawn over here. So, something very interesting happens over here. If I shift it a little bit, I got a trajectory that went like this. Can you guess why from this? Right. So, you are actually defining a threshold through this.
Yeah.
But why did the trajectory itself move this way?
Because you go along the line. It was less of the blue.
The threshold is inferred from this, so you don't use the threshold to explain it. That's tautological. Use the maths to explain why you went here
So you go,
versus going there.
go along the blue line.
Yeah.
Once you reach the yellow line
Yeah.
domain,
Yeah.
so there are you can go both ways.
That's right. So there's a little funnel here. If you get caught up by this, you get pulled by that. If you get caught up on that side, you get pulled at that, and the threshold is exactly this blue manifold, right? So that blue manifold is a separating. It's a separating plane for, or separating line, separating curve for whether you go this way or whether you go that way. So it is possible by a very small change in initial conditions to actually go back right down to the voltage, right? But if you change it just a little bit on the other side of the blue, you get there. Right? So that's a very sharp separatrix. Okay. So now my guess is this trajectory, which spent some time over here, should have a larger latency than the one we had earlier. Okay, let me see if I can, if I was right, or if I can do that. There it is. I have been able to change my latency. Right. So your stimulation came here, your perturbation happened here. You spent 20 milliseconds doing nothing, or barely rising above your baseline, and the next 20 seconds was your action potential. You waited 20 seconds, boom, then another 20 seconds, your action potential. Right. So what does this tell you in terms of the frequency? What do you think is the frequency range for this neuron?
There's a very large range of frequencies possible.
There is a very large range of frequencies possible. In particular, you can get frequencies that are low. Right? You saw that there was a band limited frequency range for the hop case. Right? Now I am asking the question whether you can get arbitrarily low frequencies. Can you have frequencies which are really close to zero, very slow frequencies? You think you can?
So the one question you asked initially, so action potentials are in milliseconds, right?
Yeah.
But how do you encode something which is over, say, seconds or minutes?
Yeah.
So that way you can.
So there is going to be a range for ionic currents to play in terms of the time scales that you can use ionic currents to explain. At some point, you are going to have to shift into the dynamics and the time scales of other molecules. So I can maybe set it up, I can maybe suggest a general sort of plan when people build models. For the fast time scales, you work with ionic currents. For slower time scales, you work with calcium.
Or maybe cyclic MP or things like
We'll get to cyclic MP also. You work with calcium. The next level of time scales, you work with mitochondrial calcium. The next level, you work with things like glycolysis, where cyclic MP and so on can enter, right. So mitochondrial dynamics is of a scale that is slower than that. The next level would mean something that's happening at even slower rates. You can stretch it all the way out to something that's changing on seasonal scales. Once you're beyond about 20 minutes or so, I think you can involve changes in genes and mRNAs and such. Yeah. Okay. So coming back to neuronal systems, now the next thing that so let me maybe just for the sake of completeness show you the direction flows. Yeah. So it turns out that this is interesting that there is this separatrix that came out from here, this unstable manifold came out of here, actually dies in this one over here. So there is a separation between these two, and then there is this our action potential trajectories look like so. Okay, so that's what this looks like. Now I am going to do the following. I am going to ask where does an oscillation come from in this, and then we will show you the bifurcation. Okay, so if I want to get an oscillation, what do you think I should do? I think I am going to inject some current. Injecting the current will do the following, it will lift this, the, so what will happen to these two fixed points? They'll come closer to each other, right? And at some point, these two will come to get close enough and then pop off, right? It will go right through. So we'll go from having three fixed points to having one fixed point. Is that plan clear? Okay, let's see. I'm going to go from 20 to 30. Let's see what happens to the alkaline. Okay, so the alkaline has shifted. The alkaline has shifted, and these 2 points which are on the outside have moved slightly in, right? And that's the saddle point, the one which had stable and unstable directions. That's called a saddle point, and the node. The saddle and the node have come closer together if I lift my alkaline. Okay. Okay, they're very close here, right? Okay, there you go. So this pair of saddle and node, they they collide into each other, and if you raise the IAP a little bit more, then you actually lose those 2 points. This collision of a saddle and node and the passage through this bifurcation called a saddle node bifurcation. That's the origin of the name saddle node, right, in SNCC. Okay, so now let's study this case to ask what happens here. Anyone guess? Now you get a cycle. Okay, let's see. Let me start here somewhere. There you go. So this guy goes all the way around and pretty much comes up over here. You can see there's a little mismatch here, but I can keep continuing it and I'll get a full cycle. So now you've got oscillations in your model. So what has happened in this model is you've lost stability of 2 fixed points simultaneously, and a single fixed point remains, and what appears in its stead is a periodic orbit. So it's as if the moment you lose your 2 fixed points, the saddle and the node, the 2 lower ones, right, and there's a limit cycle that appears at that very moment. Okay. So that bifurcation is causing not only a saddle node collision, you also have an invariant circle emerging at that point. So this is why it's called a saddle node on an invariant circle, a saddle node on a limit cycle. Okay, does that make sense? There is no resting state here.
Yeah. So it will keep on.
It will keep cycling. Okay, let's take a look at that. Let's see that. Okay, perhaps it could be. Let's start at some other point in the plane. I come to this. Any point in the plane I start with, I'm going to that. Okay. Perhaps you're not convinced. Okay, let's take a look.
So basically, stable path is there, and it will fall back into it over and over again.
Yep. No matter where you start, you're going to get this side. Okay, and there are no fixed points here. I mean, there is a fixed point, it's unstable.
Yeah.
Oh. Okay, so
also lost the latency. The latency.
but now you're putting a fixed period.
Yeah.
So I don't. So I'm.
Fixed,
I'll answer that question a little bit later. Okay. Let me go to the bifurcation diagram, because this diagram doesn't tell me what the frequency is. This diagram tells me that I have a limit cycle. I can change my current, I can increase it a little bit more, and I can still expect limit cycle. That it will tell me, right? Okay. And at some point, it'll also tell me that if I do it, if I raise my current large enough, I will stop getting the limit cycle. Right? Those are things I can guess from here, but I don't know the frequency. I have to plot the bifurcation diagram and then compute the frequency. Okay, so let's do that. Let's go back down to 20. I think we had a stable point here, right? Yeah. Okay, I have a point here. Let me get auto up, parameters I have, numerics. Okay, so here's what happens if I run a steady state bifurcation diagram. I go through this branch of stable points. That branch actually turns around, and I have an unstable branch and a stable branch forming a knee. Okay. That branch goes backwards, turns around, and then I get stable points. Okay. So, can I look for a hop over there? Let's try and see that. Yeah. This is a limit point. In fact, this little label that says LP shows you that it's a limit point. Okay. That limit point means there's a knee of this. There's an elbow. Okay. Let's go all the way up here. I have a hop point over here. So, this point right here where things change, that's a hop point. Okay. So, now I am going to do the following. I'm around the hop point I expect periodics. So, I am going to draw a branch of periodics out of this hop point. So, I have grabbed this hop point, and perhaps I should change the numerics to negative. If it does not work, we will change it back to positive. Okay. Sorry, what is that? Run periodic. There it is. Okay, what do we see? We see a subcritical branch come out of unstable periodics that turns around. We've seen this before. That turns around to branch of stable periodics, and those stable periodics die at exactly that place where you had the hop, where you had the limit point. Sorry. Have you seen this before? It's the saddle node bifurcation. When that saddle node bifurcation occurs and you go slightly past it, there's a limit cycle lying over there. So, at this point when the saddle node occurs, there's a limit cycle that emerges. This little blue stuff, that's a little bit of a numerical error, right. So, this is called a saddle node on a limit cycle bifurcation. Why does this have little blue? That's just numerical error. I can do it more carefully. I didn't want to do it right now. It will go away if you do it a little bit more carefully. It's due to a little bit of numerical error. There's a homoclinic orbit over there, which Otto is not quite able to grab. Yeah, you'll have to change the numerics a little bit to get it. Okay. I didn't want to change it too much because I wanted to use the default values. So, I let this branch be computed with a little bit of error. Okay. Does that make sense? A stable point and two unstable points, that's correct. But I just showed it to you. When I did one of them was a stable, think about it. So, if you go back and compute it at this value, you will actually see that there are, one is a saddle, the other is an unstable node. Unstable node was it? We will check.
So, usually the topmost unstable point will become the stable point as we keep increasing, like the one in the.
That's right. So, you lost a saddle node over here, this point stays unstable. As you keep raising it, this point actually moves on the right branch over there, then it becomes stable right around there. No, yeah, it will pass through a hop position over there and then it will go over. Okay. Does this make sense? So, now the next thing that we want to do is plot the frequency. Okay. So, let us take a look at this. What do you think will happen? Any guesses? This is what happens. You get a quadratic branch coming out, you can get arbitrarily low frequencies. Okay. In the hop case, you had a frequency limited band, a band limited frequency. Here, the lower frequency can be as low. That means you can have oscillations of very small, very large periods. So, in principle, as he had suggested, perhaps very long behaviors can be understood by very slow neuronal movements, neuronal bursts, neuronal firings, right? Perhaps you might use that sort of behavior, or perhaps you might use other molecules to explain slow behavior, but it is a possibility. In fact, there are models of say for example beta cell dynamics, where glucose stimulates both electrical activity and it stimulates glycolytic activity, and it stimulates electrical oscillations and it stimulates glycolytic oscillations, and there are regimes in which the two time scales interact with each other. So, there is a lot of lovely work by Arthur Sherman and colleagues, where you can study things like this. You can have couplings of different oscillator regimes. Yeah.
But to reduce the frequency, like slowly the amount of change in current required to go down a frequency becomes very small.
Yes, it happens over a very narrow range of current, if that's what you're asking. And this branch,
It might not be possible to physiologically distinguish that.
that's in a way that's correct. So, this region though, this branch, it can be shown that this is a quadratic function, okay. And in fact, there are models of neuron, caricature models of the neuron, in particular that call the theta neuron, that rely on a quadratic description here. So, one works with this regime alone to build models of interesting type.
This amount of current is achievable, this low.
Yeah, absolutely.
No, but the distinction in
Oh, that's just because you're comparing it. That's because you're comparing it on that scale. If you operate with the current on that scale alone, he says that is not unreasonable. Okay, so this is what I wanted to describe to you in terms of the neuronal properties that can differ. So, I have gone through all of this. Let me just show you that these are called class 1 and class 2 neurons, and the properties are distinguished as follows, right. So, class 1 are snake neurons and class 2 are hop neurons in the sense that I have described to you. In class 1, axons have sharp thresholds, right, can have long latency and can fire at low frequencies. So, I urge you to go back and make sure that you understand through all of this analysis that we have done where these properties come from. Okay. I have shown you all of that, but make sure that you understand it. In class 2, axons have variable thresholds, short latency, and there is a positive minimal frequency. It is band limited. The frequency of this neuron is band limited. Okay. So, that is the difference between class 1 and class 2 neurons. So, Rinzel and Ermentrout were the first to note this connection, that these two different classes of neurons, the description was by Hodgkin in the 1940s. The correspondence between the Hodgkin classification and properties in terms of bifurcations was first noticed by Rinzel and Ermentrout, and this is now a staple in the field of neuroscience. Okay. So, as Bard also suggests, I will leave the third class as a exercise to you for those of you who might want to study it as a project. Okay, so that is what I wanted to say about types of neurons. Let us see where we are. So, by looking at solutions alone, you do not get quite a full picture. Oftentimes switching to the phase plane is very valuable. You get null lines out of it, you get direction fields, you get flows. We have gone through all of this to see what sorts of wonderful information these geometric structures can bring for you. You go to the bifurcation diagram from that point on, and then you can get the frequency response of that neurons. Okay, so that is what you can do with these things. Now, I will maybe take another 5 minutes or so. If there are any questions, let me first take that, and then maybe I will show you a few other things to give you pointers for future directions. Any questions? Yes.
So basically, in the two models, the two nucleus that you for the hop application of the snake, it was essentially like the red was shifted left to create. So there was, and So
the injection of current lifts the voltage in alkaline or depresses it.
correct, correct. But between the two of them,
Hmm.
so the first set of graphs and the second set, there was like a lateral shift.
So the relative orientation
Was
is, yes, the relative orientation matters. So when you have, roughly speaking, when you have an intersection like so, you get a fixed point over here. When you have an intersection like so, you'll get an oscillation, and when you have an intersection like so, you'll get frozen at this fixed point.
so, but between the two of them, we'll have like both the like bifurcations happening together.
Right. So this is the fixed point where you're frozen at a high level. This is fixed point when you're at the resting level. Here you've got an oscillation. So this is the going between this and this in terms of current will give you the bubble.
Right, right. Okay. So,
So I wanted to make one point before I wanted to make two quick points. If there are no other questions, if there are, we can tackle those. Okay. So these diagrams can also be used to study what are known as slow fast dynamics. Okay, Siddhabra mentioned it a little bit. Let me describe this a little bit more because this is a very nice, interesting aspect of neuroscience to study. Okay, so you will see, you've seen that this voltage in alkaline is, this is the voltage in alkaline, and this is the and then there is a N alkaline. Okay, so oftentimes one studies singular versions of these. What that means is the following: when you see an alkaline picture like this, you will see the trajectory come along like this, move like that, and go around like that. Sorry, I shouldn't have overlapped my trajectories, right? Like this. Okay, this corresponds to the oscillation that we are familiar with, right? You can, if you change the time scale separation between these two, right, which means that one of these is happening at a very fast scale and the other one is happening at a very slow scale relative to the other one, and I'll leave it as an exercise for you to figure out which one is slow and which one is fast. You start to see the following: this loose loop becomes tighter and tighter relative to these alkalines, and in a sort of limiting case, you get the following: the jump off is fast and at the knee, you move, you hug this alkaline, you sort of cross this alkaline and you're sticking close to it. So effectively in the singular case, you're actually moving on it. Okay. If anyone's interested in how this happens, feel free to contact me afterwards. I'm giving you a suggestion of where you can go with these things. Then you'll jump off at the knee. Again, this will be a fast move. And if you're in the non-singular case, you will cross it and you will hug it. But if you're in the singular situation, you will actually move along this at a slow rate. And that was the origin of the cycle that Siddhabra drew. Okay. This is called a relaxation oscillator. Okay. So a lot of things that one does is to study relaxation oscillators and what sort of patterns can emerge from relaxation oscillators. The relaxation oscillators are the limit of studying things like Morris-Lacquard in the singular limit, what's called the singular limit. The singular limit corresponds to separating the time scales of the two variables to a very large degree, and in that limit it becomes the relaxation oscillator. So you can study things like post inhibitory rebound. I have run out of time, so for those of you who are interested, I can show post inhibitory rebound, or maybe Siddhabra can cover it at some point using relaxation oscillators, or I can give you the position in Bard's book where you can study it. Sorry Siddhabra, I thought I would be getting to it, but I ran out of time. So yeah, actually it's a nice exercise. I'll show you where it is covered in Bard's book. And so this entire chapter is called fast flow analysis, which deals with the limit of the Morris-Lacquard in this sense. Okay. So it's a lovely chapter. You can get as deep into it as you want. Beautiful mathematics over here, beautiful stuff to deal with, right? And lovely set of papers written by David Terman and others and his colleagues. So you can study those sorts of things. Let me just leave you with the diagram of post inhibitory rebound, and you can try to figure out how that goes. Yeah. I'll come back to this in a minute. So that's the stuff that I was trying to describe to you. Jump up and jump down. This diagram shows you post inhibitory rebound. Okay. It's a nice from a geometric picture alone you can understand what goes on, and all of the tools that you need to understand PIR I have described to you already. So it will be a nice exercise for you to work out what PIR is. Okay. The next thing that you can try to do is to take relaxation oscillators and start coupling them. Okay. The coupling equations we have already seen before. In fact, they become even nicer, even simpler in the sense of when you start coupling relaxation oscillators. Okay. So, in this very chapter, a few pages around, you will be able to see couplings of relaxation oscillators, and then you can study networks of relaxation oscillators, right. So, now I hope you have a sense of what are some of the things that are covered in this book and what are some directions in which you can do your projects. You can consider networks, you can study things like synchronization, you can study things like half center oscillations. So, for example, if you look at this diagram over here, was it this one? Yeah, this one. So, for example, he's coupled two neurons together, one is in dashed and the other one is in bold, and you can see that one neuron fires, the other neuron fires, and the first neuron fires and the other neuron fires, and they sort of do go in and out, right. So, that is a half center oscillation. Exactly what do you think would be the kind of coupling that is needed to give you half center oscillations? And you can ask questions like this was the uncoupled oscillator before it was connected into a network over here. What is the relationship between that frequency and the frequency of the neuron in the network? Would you agree that the frequency of the network in the network, the frequency is actually lower of this neuron? So, how can you connect a neuron such that in the coupled sense the neuron is now firing at a lower rate? Would it be coupled with excitatory or inhibitory synapses for the neuron to slow down in its frequency?
So, if the neuron is facing up actually, facing up, frequency increases.
Okay, good catch. But the frequency of the single neuron in the network of neuron 1 is actually lower, I think.
No, you see, the time scale is same, and the peaks, number of peaks is higher in the bottom.
Okay, it could be, yeah. So, the point I am trying to make is you can study patterns of this type because these are very important in how neurons, how a group of neurons could be talking to each other. For example, Siddhartha mentioned a group of excitatory neurons and a group of inhibitory neurons. If you were to study the dynamics between these two, what kind of behaviors can emerge from this? Nancy Coppell, for example, has a lovely body of work talking about what are called ing and ping networks. So, these are pyramidal interneuron networks, and she can show, she and her colleagues have shown nice set of oscillations in the alpha range, in the beta range, in the gamma range of frequencies. So, another direction for you to study, look up the old literature, try to work out what kind of rules you can work out for how these networks couple together and how they behave in networks. So, there are lots of emergent phenomena here, right, because what you can't understand by just looking at one neuron, what you can understand by looking at one neuron may differ from what happens to that neuron or how it behaves when it's coupled in a group. So lots of scope for studying emergent phenomena. So I think I'll stop here, and if you have any questions, you can catch me later today or over email or Zoom when I'm back. Yeah.
I have actually shown them a simulation post-COVID.
Go ahead.
No, I mean,
Yeah, no, go ahead. It's up to them if they want to see.
so basically, like the equation code that I sent you, I will actually after lunch, if Sumitra gives me some time.
She's not here, so she's volunteering.
She gives me some time, I can just show you on the code how to get post-COVID.
And then you guys can try and work out if you can understand from a geometric point of view by studying the phase plane alone. If you don't get the answers, look it up in Bart's book.